Our next final speaker is Dr. Christian Schafmeister, who is an associate professor at Temple University in Philadelphia in the Department of Chemistry. Prior to his appointment at Temple, he was at the University of Pittsburgh. He received his PhD in biophysics from University of California in San Francisco, and completed postdoctoral studies at Harvard under Professor Gregory Verdine. In 2005, Dr. Schafmeister received the very prestigious Feynman Prize for Experimental Nanotechnology. His research interests include the areas of chemical biology and nanoscience, and as he will illustrate during his presentation entitled Using Molecular Lego for Nanomedicine. And again, this is another fascinating, very different story on the delivery of medications using nanotechnology. Christian.
Thank you. Yes, this is going to be very different from what we've seen so far. Thank you for inviting me.
Sure.
Let's see. Do I control this? I guess that's a general question. All right. So I'm now a synthetic chemist. I was trained as a biophysics. That's what I did my degree in, protein design, X-ray crystallography of proteins. But for the last 10 years, I've been a synthetic organic chemist. I had 100 students running my Organic 1 exam 2 days ago. And so I'll show you what we've been working on the last 5 years. The basic idea is to try and make molecules that can do the things that proteins can do. All right, proteins are these wonderful structures that can catalyze chemical reactions, build structures. They are the molecular machines that make life possible, and I really want to make things like that. And originally, I tried to do that by making unnatural protein. I made one of the first unnatural proteins. It's called 4-HB1. The crystal structure is in the PDB. The thing took me 4 years to put together, and it's a molecular boat anchor. It doesn't do anything, and I know how to make a 4-helix bundle, but I didn't know how to make anything interesting like this, like an enzyme. And so I thought, I don't like folding. I'd like to do something else, come up with another way of making big molecules that can position functional groups, but that are much more engineerable than, proteins, which are like Jell-O. So there are other people who figure this thing out. There are other people who have working on this idea, and this is the general area of foldamers, make new building blocks that are easier to, that fold more readily into three-dimensional structures. And they can primarily make helical structures. I had a different idea. I thought, what if I make building blocks, and instead of making them so they couple through single bonds, like DNA, proteins, carbohydrates, let's make them couple through pairs of bonds. And let's make the building blocks cyclic, so that they have, the core is some ring structure, and then we'll attach two groups on each end of it, so that when we connect them to other building blocks, they will connect through pairs of bonds, and you make a rigid ladder molecule. And then you can use this ladder as a scaffolding to present functional groups the way proteins do, but they don't fold, they just have shape because of the sequence and stereochemistry and structure of the rings. So this is what our building blocks look like. They are bis amino acids, what we call them. They have a cyclic core. They have an amino acid on one side, another amino acid on the other side. Every building block has two stereocenters. You have to synthesize them absolutely stereochemically pure, and you have to be able to make gram quantities. So we've solved all these problems. It costs less than $20 a gram to make this building block. That's starting materials, solvents, silica, everything. And there's only one column involved in the synthesis. And we have lots of these. We have 5-membered rings, 5-6 fused rings, 6-membered rings, different regioisomers. We can make every stereoisomer, 4 of them, of this building block, every stereoisomer of this, 2 of this. We can turn them backwards. We have an enormous amount of backbone control. What we figured out how to do in the last 2 years is how to put functional groups on these things, like amino acids have side chains, and then get the coupling chemistry to work, because that was the problem. Putting functional groups is not the problem. Getting the coupling chemistry to work is the problem, because everything shuts down. All the reactions shut down. We invented a new reaction that solves this problem in a beautiful, elegant way. So now we can make molecules like this that have a rigid backbone, and they present a functional group on every building block. We can control the distance and the relative orientation of these building blocks by controlling the sequence of the monomers, the structures of the rings, and the stereochemistry at all these positions. It really is like a molecular Lego set. Here are the parts, and we can stitch them together in a couple of hours and days to make molecules of almost infinite variety. The way we control shape without folding is like this. This is a chain of bis amino acids. It's a ladder molecule, a spirolateral ligament. And we can go onto one of these monomers and put up all these black lines. These represent all the different directions the chain could turn if you put one of the other building blocks that we have in this position. So you put another building block in, you click this line, puts it in, now you have a hairpin turn. You can go to any other monomer and do the same thing. So we can basically make molecules that follow any path through three-dimensional space you want. It's a single continuous chain, it doesn't fold, it's a ladder, bent, weird, twisted ladder, and we can decorate it with functional groups. We've made more than 100 of these molecules. We're just now starting to develop applications now that we can put a lot of functionality on them, but we've made macrocycles and all sorts of different shapes. We've published about almost 20 papers on this, and we're the only group that's working on it. This is the only thing we do. So one application that we developed was to make molecules that make very large conformational changes, like myosin. So you have a molecule that's got a 2 nanometer rod, a hinge, another 2 nanometer rod, and then put 2 binding groups on the end. And the idea is that when it's unbound, it's floppy and large, and when it binds something, it would close up into a more compact shape. We could use this for all sorts of applications, like sensors. You could put these on nanotubes and put a charge up here. When they bind their analyte, they'll fold over, bring the charge down to the nanotube, and you've got a chemical field effect transistor. We could put these on the surface, and we could detect binding by changes in height with atomic force microscopy. We can put these in nanoscale channels and make valves, active, smart valves that recognize something. If we put boronic acids on here, you add glucose, the glucose binds, folds the thing up. Now you've got a channel that insulin could flow through. You could make a material that acts like an artificial pancreas. So this is the molecule that we made. This is Laura. She's a master's student in my lab a couple of years ago, and she made this molecule, very easy to synthesize. And we put metal binding groups, so it binds copper and folds up. You get very nice binding transition as you add copper. And we use biophysical techniques to characterize these. So when it's open, you use sedimentation velocity experiments, and it falls very slowly through the liquid when you centrifuge it. You add the metal, it closes up, and it falls faster. It's like a terminal velocity. With a parachute, you fall slowly. With a parachute closed, you fall very quickly. And we can actually use these numbers to figure out what the axial ratio, the size of the molecule and shape is. And you can see that it goes from a long axial ratio to a compact molecule. Also, if you put it in a size exclusion column, when it's open, it comes out early because it's going around all the little holes in the beads. And when it closes up, it becomes more compact and it gets lost. And it takes longer to get through. So its retention time changes. Now this idea of metal binding with all these, now that we can put a lot of functional groups on, we can make molecules like this. And I've got a student, a postdoc, who's working on synthesizing this molecule. It displays 3 imidazoles to bind to zinc and leave an open coordination sphere so that it could bind carbon dioxide and perhaps be used to convert carbon dioxide into carbonate like carbonic anhydrase. And this could be used to capture carbon dioxide for sort of clean coal technology. These things are robust. They do not denature. You could boil them and cool them down, and they're perfectly fine. They're rigid ladder molecules. This is another application we're designing to make a broad spectrum antibiotic by making a triangular molecule that's about 2 nanometers on the side. It's got a hydrophilic core, and these rods are several of our bis amino acids connected together with a hinge on each corner. Put positively charged groups around the outside, and then a gasket of greasy tails. And I think what this thing will do is associate with the anionic membranes of bacteria and then dip down into them and create holes where the bacteria could leak their guts out. This is the principle that our own antimicrobial peptides that our bodies are synthesizing all the time use to kill bacteria. But these things could work at 1/1000th or 10,000th the concentration because they don't have to self-assemble. They're pre-assembled synthetically. So they could work at very low concentrations and be cost-effective broad-spectrum antibiotics. We have a grant from the DoD to develop these as artificial enzymes. So in collaboration with Ken Houck at the UCLA, he designs transition state models, computational models of lowering transition state energies of reactions like this ester hydrolysis, and figures out what functional groups you need in order to accelerate the reaction, to lower that activation barrier. So then what we do is look for bispeptides that can hold the functional groups in space, the same way an active site of an enzyme does, but a tiny little scaffold. And then we are currently developing several of these, but we synthesize the molecule and then we throw the substrate at it and we see if it catalyzes the reaction. If it does, it'll change a lot of things. We have grown crystals of these. These are not diffraction quality, sadly, but we are still working on getting diffraction quality crystals of a bispeptide, but we've done lots of NMR structures. So we could take the structural information from these and feed it back into the design and hopefully one day rationally design catalysts. This is one reaction that we're going after. It's an aromatic Claisen rearrangement, and what happens is that you develop a negative charge on this oxygen. And so Ken's group has modeled active sites like this, where you have two hydrogen bonding groups that stabilize this negative charge. And so we are making molecules, very simple molecules like this, that hold a carboxylic acid and a phenol to simultaneously hydrogen bond to that oxygen, and then test them for their ability to accelerate the reaction. That one, the first one that I just showed you, updated, this one doesn't work, sadly, but we're making other ones. We believe that this is forming an internal hydrogen bond that would have to break, and so we're making other versions that pull them apart a little bit more so that they cannot form an internal hydrogen bond, and see if that accelerates the reaction. And then more complicated versions, this is a model of a serine esterase that Mubube in my lab is making, where you've got an alcohol, an imidazole, and a carboxylic acid, and then an oxyanion group, and all laid out with the right stereochemistry and structure of everything so that they're held in a line to mimic an active site of a serine esterase. This is the most advanced project that we have. The idea is to make new drugs, so molecules that can bind protein surfaces. So a lot of proteins bind helical peptides, and what I thought is, okay, what I'll do is I'll look for bispeptides, our molecules that can present three groups to mimic the side chains of an alpha helical peptide. And so I've written a lot of software. It's about 150,000 lines of C++, runs in parallel, builds these things very rapidly and scores them for how well they can present functional groups in precise orientations. And this is one of the stereoisomers that it found. Here's a superposition. This protein is HDM2 or MDM2. And it's binding the p53 peptide. p53 is the policeman of the cell. One of the ways that about 50% of all cancers suppress p53 is by expressing more of this protein. So if you can make a small molecule that binds to this protein, you'd release the green peptide, the green protein, and it could then initiate apoptosis or cell cycle arrest as an anti-cancer therapy. This has been demonstrated with small molecules. So we're trying to make this. We've made this purple molecule that can bind in here as well. And the software actually generates lots of stereoisomers that can hold three groups in the right orientation. So we've just started synthesizing these. The first thing we did was start with one stereochemistry, one backbone shape, and then screen different functional groups that could get binding. And we got binding right away, but it was pretty weak. But we got down to about 5 micromolar before we picked a set of functional groups and we said, okay, we're going ahead with those to do something new. And that is to vary the stereochemistry of the backbone to sort of adjust the fit of these three groups to see which ones can best bind into the binding site. And so we went from about 23 micromolar with this stereoisomer down to this one here, which is now 400 nanomolar, tighter than the natural peptide. And all we did was we made 20 compounds. Right? Contrast this to the small molecules. You screen libraries of like 300,000 compounds to come up with one sort of low micromolar inhibitor lead compound. We found something pretty quickly with this approach. These compounds appear to get into cells. They have a fluorescent tag on them, and they're about 1,200 Daltons. So we mix them with liver cancer cells, and then after 24 hours, wash the cells out, and they're all glowing green. These are cells with mutant P53. These are cells with wild-type P53. This is a confocal image of some of these mutant or wild-type P53 cells with our compound, and the compound is all throughout the cell. So they're getting in. I'm not sure if it's active transport or passive diffusion. Passive diffusion would be very exciting. This is very new, and we're just following this up. But we did notice that with these wild-type P53 cells, there were a lot of dead cells that have come off of the surface. And I know it's tantalizing. We're excited, and we're following up on this to see if these compounds are getting in and activating P53. And then where we're going from this is we synthesize the building blocks individually. How much time do I have left? I think I'm way ahead of time.
Oh, yeah, you go.
Oh, wow, I talk too fast.
15 minutes altogether.
Oh, okay, well, I'll be done very quickly and we can get on. We can synthesize the building blocks and we can put functional groups at other positions as well, not just on these nitrogens. And so then we can assemble those pieces back together again. So we can display a lot of functionality on this backbone. All these different R groups are places where we can put functionality in. And we've published synthetic approaches to all of these guys. And so we've started screening this functional group. It's the easiest one, R4. And we screen this one, and we see that we're getting, sub-micromolar binding with different stereochemistries. And we've got a lot of work to do here, a lot more compounds to make to just see how far we can go with this. And that's basically it. This is my current group. They're all working on different applications for these molecules now that we can make them and put a lot of functionality on them. We're developing applications. I've had funding from the NIH, from the NSF, and now from the Defense Threat Reduction Agency, DoD, and Temple University. And with that, I'm done. I thank you, and I'd be happy to answer any questions.
Other questions for Sam?
Oh, yes.
So Molly Schweik at U of T. Do you have any issue with solubility? No, none at all?
These are wonderfully soluble in water and polar organic solvents, and as we put more greasy groups on them, they become more and more soluble in non-polar solvents. I think it's because they have their rigid structure breaks up their hydrophobic patches and their hydrophilic patches, and they're frustrated because they can't adopt different conformations to become self-complementary. There's only been one case where we tried to make molecules that could hydrogen bond with each other. That stuff was brick dust. Everything else is very soluble in water acetonitrile, chloroform, DMF, all sorts of solvents.
Really nice talk. Really
Thanks.
interesting. Jay Hickman, UCF. Have you tried to apply the software technology you're using to design these things to look at A beta and look and see how A beta forms oligomers?
A beta is you mean?
Amyloid beta, it's the peptide in Alzheimer's disease.
So the software that I have is what it's doing is it's taking these rigid building blocks and then connecting them together, applying the matrix math to figure out where building block I+1 attaches to I. It can do this with amino acids, but amino acids are so conformationally flexible that you have this enormous conformational space that you need to search, and it becomes a protein folding problem, which my software could tackle, but it's really focused on what I tried to do here was design monomers that are more computationally tractable than amino acids. And the software can do amino acids and sugars and anything else, but we're tackling the bispeptides. Yeah.
Very nice talk. Shui Li from MCG. I have a quick question. One of your slides showed that you labeled your peptide with fluorescent for the labeling. My question is, after you label this fluorescence, do you saw any interference with your protein or peptide function?
Actually, we think it's involved in binding the protein because there was one stereo center that we had set one way initially to lift it away from the protein if those three groups are binding into the pocket. And then on a LARC, Zach flipped that stereo center, which would push it back into the protein. The binding improved. So there's hydrophobic surface up there to make contact with, and we think that the fluorescein might be binding, contributing to the binding now.
Okay.
And we're just now taking it out and looking at it in competition experiments to see
Yeah,
how much.
The reason I'm asking is that we are trying to monitor the protein trafficking through the membrane or to the nucleus. We found that once we label something with fluorophore, it will interfere with the behavior of protein.
Yeah.
Okay, thank you.
So, Stephen Lee, OSU. I wanted to mention something that you mentioned really only in passing in your talk, but it's potentially very important, and that is this is one of several nanoscale antibiotic materials I've seen people propose, and most of them work by somehow interfering with membrane function. And the thing that's potentially important about them is if you go back and study the literature of plasmid biology and you look at where antibiotic resistances come from, they mostly come from genes that were present in the synthesizing organism that subsequently end up on an X chromosomal element and are transmitted horizontally. Right? Whereas these sorts of compounds have the advantage in that there is no synthesizing organism except you. And so therefore the possibility is that resistance to these things may be much slower kinetically to develop in the population than conventional antibiotics. And this is important because I think we're in the process of returning to the pre-antibiotic era. And so infectious disease was a major cause of morbidity and mortality in the past. Having new antibiotics certainly would help us to avoid going back to that state.
Yeah. Well, our bodies right now make antibiotics like that. They're called antimicrobial, cationic
Like defensins
antimicrobial.
and meganins.
Yep. And bacteria have not evolved much of a resistance over the millennia. The feature that those peptides are exploiting is that bacterial and membranes are primarily negatively charged on the outside, while human cells and mammalian cells are neutral on the outside. So cationic peptides electrostatically stick to these membranes. And, we're making them all the time. They're protecting us. They're part of our ancient immune system. Insects don't have T cells and antibodies. They have primarily
Mombicin,
antimicrobial
things like
peptides,
that.
and they do very well with them. What we're trying, the problem with them is that you need a fairly high concentration because they have to self-assemble, and they're only effective, the bottom is maybe about 1 micromolar to 10 micromolar. So they'd be very expensive to use they're very expensive soaps, basically. But if we can pre-assemble a sort of a portable whole that could drop into a membrane and not have to self-assemble, they could work at very, very low concentrations, and that's why it's sort of a nanotech approach to antibiotic.
The point I was making is T1 have to go from introduction of a new antibiotic to prevalence of resistance is now like 10 to 15 years. It's not very long. So the functional life of the antibiotics are limited. There's a rational, reasonable reason to hope that things like what you're suggesting might actually have a longer, useful clinical
Yeah,
life.
I hope so. Thank you.
Yes, it's not really a question, it's more like an idea. I don't know if you've ever heard of the work of the Demain, Eric and Martin Demain. There are two MIT professors, and they work on origami, molecular origami. And there is a lot of work done with DNA, which has a lot of problems. Well, your stuff, especially the one that you were showing at the beginning with the molecular hinges, it's something that really recalled me of that work. Have you ever considered to do anything in that direction? Because there are a lot of applications in the nano world for molecular origami.
Actually, I think I know how to make protein-like objects and complex asymmetric materials, because we can put metal binding groups on these things and do metal templated assembly. We're starting to do that now. To build three-dimensional structures. We can also make short oligomers and then chain them together like building blocks. So we can make protein-like structures that would be kind of like segment, slats connected by hinges with appropriate functional groups so that they could fold up, but like a child's puzzle, with lots of complementarity on each subunit. And we're working on that too. It's synthetically, I totally see how to do it now, and I've got people working on it. Sure.
What do you mean childlike puzzle? Ones that are,
Well, a kid's puzzle has big pieces.
Right.
And lots of, it's obvious how they fit together. A
okay.
adult's puzzle has thousands of pieces and they're tiny. I think the protein is like a adult's puzzle, and what I'm trying to build is like a child's puzzle, easier to put together. My 5-year-old would appreciate that.
He'll be helping, I imagine.
Yeah.
Well, thank you very much for that excellent final talk.